Okay, so today we're going to be talking about cell signaling. So we've talked about the membranes and all of the different proteins that are within those membranes. And so we want to talk about how cells can talk to one another. And there are three components of cell signaling. First, there's the perception. So in other words, it has to perceive that there's a change in their environment, or we also call that the stimulus, so perceiving the stimulus. And then secondly, there is the signal transduction. So once we've perceived it, we need to send a signal to the part of the cell that can then respond to this stimulus. So of course, the third component is the response. Works a little bit like telephone, right? We have our perception of that information coming into our ear, then we respond to the stimulus. Well, within cells, our perception oftentimes happens at the plasma membrane. So at our plasma membrane, we have specific proteins that are receptor proteins. And usually a signaling molecule will come in and it binds specifically on that receptor. The receptor will have a particular site for binding that molecule, and that signaling molecule is often what we call a ligand. That's the terminology. Once the ligand binds the receptor, then there's what we call transduction, and that's just basically a relay of information, and that can happen in several different ways. And so we call these either relay molecules, or we can also call them secondary messengers. But at the end, the last secondary messenger then causes a response, and that's an activation of cellular processes or a change in genetics. Okay, so let's go back to that signaling molecule. And like I said, that is called a ligand. And this definition of a ligand would be a molecule that binds specifically to another molecule that's often larger than itself. And that causes a change in the shape of the receptor protein that it's binding to. And we often say that this is the lock and key principle because very much like enzymes and their substrate, there's a specificity to that activation site. So the ligand has to be a specific shape, have specific charges in order to bind the receptor. Okay, so what about these receptor proteins? Typically, they come in two different varieties. The first is a G protein coupled receptor, and the second is a ligand-gated ion channel. So let's take a look at our G protein coupled receptor. So this receptor is again going to have a specific binding site for the ligand, or our signaling molecule here. And the G coupled protein means that there is a G protein literally attached to our receptor. And when our ligand is bound, then that G protein becomes activated, and it's a G protein because it binds and is activated with GTP. Similar to ATP, ATP is adenosine triphosphate, GTP is guanine triphosphate. So once that G protein is activated, it then usually slides over within the membrane, because remember it's a fluid mosaic membrane, and it will bind another protein, oftentimes a kinase. And once that G protein is bound to this next enzyme, because of the GTP, this enzyme then becomes activated, and usually by using up the GTP on the G protein. And once that enzyme is activated, then there is a series of cellular responses, some transduction of that pathway. But it starts here at the G protein coupled receptor. Another type of receptor is a ligand-gated ion channel. And it's exactly what it sounds like. So we have a transmembrane protein that is our receptor protein. Again, it has our ligand binding site. And so when the ligand is not bound, that channel is closed. But once the ligand binds to the receptor protein, then the channel is open, and that allows for the passage of ions to flow either into or out of the cell, depending on what the ion channel is formed for. And once it does, typically those ions then become those secondary messengers that will allow for other cellular responses to occur. And again, once that's completed and the ligand is released, then that channel protein is available for another ligand to bind. Okay, so our second step in the process then is signal transduction. So we've talked about two different types of receptor proteins. There are others, and there are certainly other ways that stimuli can be perceived, and we're going to talk about that in a little bit. I'm going to give you a couple of examples. But these are kind of your standard, most cells use this type of process. Okay, so now we've had our receptor molecules perceiving the change in their situation. And so how do we get that information to be transmitted to the area that can respond? Well, there are two main pathways that are used. One is a phosphorylation cascade, and the second is using secondary messengers that are either small molecules or ions, like we talked about with the ligand-gated ion channel. Okay, so let's look at our phosphorylation cascade, and this is just one example. So as our signaling molecule or our ligand is bound to our receptor protein right here, then that activates usually this receptor molecule to phosphorylate another enzyme. In this case, it's the inactive protein kinase 1, which then uses ATP and is activated. So now we have an active protein 1 kinase, which can then activate a second kinase, which is protein kinase 2, again phosphorylating this one. So now we have an active protein kinase 2. And so what happens after that is there is an inactive protein here that, using the ATP from the active protein kinase, in itself can be activated. And so when that occurs, this active protein, for instance, it may be something that active protein can then go and bind a particular strand of mRNA, for instance, and allow for a new protein to be produced. So this is what we mean by a phosphorylation cascade. That just means that we are taking ATP essentially and moving it from one kinase, one protein to another, and activating it along the way. Okay, so let's talk about using secondary messengers. In this particular one, we're also looking at the G protein coupled receptor. So our first messenger protein, or our ligand, is going to bind the G protein coupled receptor molecule. And when it does, it activates this particular protein, and so then it binds the G protein to GTP, activating it, which allows that protein to then bind to this second protein, which in this case is adenylene cyclase. So just to give you an idea what we're talking about here, if you have an EpiPen, this is what it's going to look like at the cellular level if you ever have to be injected with it, because here we have epinephrine is one of our ligands in this case. Okay, so we've activated adenylyl cyclase. Sorry if I said that wrong the first time. And so then the adenylyl cyclase then hydrolyzes ATP and converts it to cyclic AMP, which is a secondary messenger. And then our cyclic AMP activates yet another protein, in this case the protein kinase A, which then through a series of other proteins eventually leads to other cellular responses. And when you take that in concert, oftentimes it means the production of certain molecules in that cell that can then be carried out to other parts of the body that allow, for instance, your airways to expand and allow you to open, have adrenaline, and other things like that. But at the cellular response, this is where that starts. So these secondary messengers are what allow for the signal transduction pathway. The third portion of our cell signaling pathway is the response. And that kind of comes in two different forms. One is the regulation of transcription. Remember, transcription was taking our messenger RNA and making proteins. And so that can either be activated or it can be turned off in response to this series of transduction events. The other thing is various cytoplasmic activities. So for instance, if we have an enzyme that's active, it may be that our signaling pathway leads to the last particular protein, then is an enzyme inhibitor, for instance, and that would stop the activity of that particular enzyme. So our response is generally a regulation of transcription or a change in cytoplasmic activity. So let's look real quickly at the regulation of transcription. So in our particular case here, we're looking at growth factor this time, and there is a receptor that the growth factor binds, and that initiates a phosphorylation cascade. That's the transduction portion of it. So once the last kinase in the cascade is activated, it can then go through the nuclear membrane, and it activates what we call a transcription factor. And that transcription factor then gives the go-ahead for the DNA to transcribe or produce a particular protein based on that particular gene. So the resulting synthesis of a particular protein is then made in response to the growth factor being perceived. So far we have been talking about the signal transduction pathway within a particular cell, and that kind of signaling is called the autocrine response. A cell is targeting itself and responding itself. There are other options for signaling, one of which is signaling across gap junctions, and that's something that we've seen that often happens particularly in nerve cells when we go across the synaptic cleft. There's what we call paracrine, which is a cell is targeting a nearby cell, so it can have an influence next door, kind of. And then there's also what we call endocrine responses, where a cell targets cells that are distant or down, like further away in the body. And so when we think of that with human body, we think of through the bloodstream, and we use the endocrine system, which usually means hormones. Hormones are proteins or molecules that are created in one particular location but are active in another. So that's an endocrine response. In plants, you also have endocrine responses. They obviously don't go through a bloodstream, but they can go through, plants have what we call a vascular system. Sounds familiar, right? Doesn't carry blood, but it does carry molecules to a distant location. So we're going to talk about two endocrine responses in plants and give you a couple of examples of that. So let's start with an autocrine response in plant cells. And yes, I know my examples are always plants, but it's what I know best. So you wouldn't want me talking about something else. Okay, so there are particular cells in plants, particularly on the back of leaves, called guard cells. And these guard cells are, in this image, these little kidney bean shaped looking cells. That is really the shape of them. And their job is to allow for water and air to pass through the leaf. And so when you want your opening between the guard cells, which we call the stoma, which is basically the hole between those two cells, when you want that to be open and you want water to go out, then you need to make those particular guard cells turgid. Remember that term? It means full of water, so that they will fulfill that full kidney bean shape. When you want the stoma or those holes to close to keep water from going out, then you let the water or the turgor pressure within the guard cells to go down. And that's accomplished by how much water is in the vacuoles of those cells. So an autocrine response in these guard cells is those plant cells' response to drought, for instance. So the perception is induced by drought causing a hormone called abscisic acid to bind receptors in the plasma membrane of the guard cells. So that's our perception step. Then we have the transduction. Once ABA is bound to the plasma membrane, calcium ions enter, signaling the potassium to leave. And that flow of potassium ions is followed by a flow of water out of those vacuoles, out of the guard cells. So that is the response. The response is the outflow of the water, allowing those guard cells to lose turgor pressure and close. So this is an autocrine response in response to drought conditions. So the perception takes place in the same cell that the response takes place in. Okay, let's start looking at the endocrine responses in plants. Remember, that's when our perception occurs in one area and the response occurs in another area. How does this happen in animal cells? In animal cells, we have the nervous system and we have hormones as well. And those two things work together in order to send information from one part of the body to the other. In plants, we have a similar system. We have a vascular system in plants as well. Clearly, it's not blood, but it allows for hormones to be produced in one area, the area of perception, and then those hormones are then transported to the areas that respond to the stimulus. So our first example of an endocrine response, or it's also called a systemic response, is what we call phototropism. So that word tropism, in plants, that means differentiated growth in response to a change in stimulus or a change in environment. So when we say phototropism, that means differentiated growth in response to a change in light. And so we're going to start out with that. Okay, so here we are, phototropism. You all have seen this before. If you put a plant in a window, naturally the shoots, which means everything above the ground, are going to grow towards the direction of light. And we call that positive phototropism. So in other words, the differentiated growth is towards the stimulus. If it were away from the stimulus, that would be a negative tropism, but this is a positive tropism. Okay, so how do the plants detect light coming in from one direction? Excuse me. Okay, so remember I said that not everything is a ligand-gated receptor or ion channel. In plant cell membranes, they contain a protein that is what we call a photoreceptor. And so you have to know that plants respond to blue light specifically, and blue light is between 400 and 500 nanometers. That's the frequency. And so these proteins that are photoreceptors, they are a large molecule and they contain a pigment called phototropin that responds specifically to blue light. So when blue light is present, the receptor molecule is going to be this photoreceptor with phototropin in it. So this is kind of a diagram of what that looks like. So the blue light is detected by the phototropin portion, the pigment portion of the photoreceptor in the plasma membrane. When that blue light is detected, there's a change in shape of that photoreceptor. Remember we said that often happens with receptors, that they get a shape change when the stimulus is present. So there's a shape change in the photoreceptor, and when that happens, that allows for a transfer of a phosphate group from ATP to the photoreceptor protein. And then that starts a transduction pathway. And based on what we know from what we talked about earlier, it's probably a phosphorylation cascade, right? Because we've already started with a phosphorylation here. So we've had our perception on the plasma membrane with the phototropin protein phosphorylating, and so then there is a cascade of events, which is our signal transduction pathway. One of the things that occurs in our signal transduction pathway is the activation of proteins called PIN, capital P, capital I, capital N. You don't need to know what that stands for. What you need to know about it is it essentially allows for the export of a hormone called auxin. So auxin is a plant hormone. And when the pins are activated, they are activated on one side of the cell so that we start to release auxin in one direction. And as you can see from this diagram, once it's released, auxin then begins to travel across the stem, and you'll notice then that the concentration of auxin on this side of the plant is much higher than on this side. And in our diagram here, this is the side that has been exposed to light. In stems, or in shoots as we also call it, anything above the ground is part of the shoot, auxin induces cell elongation. So if we look at differential growth, which is what we're calling the cell elongation, as we notice, we have the distribution of auxin being different on one side than the other. And so when that happens, in our particular diagram here, this side would have less auxin and this side would have more. And so it begins to change the shape of the shoot itself. So less growth here, more growth here. And that causes an overall response of growth like this towards the blue light. So how does that happen at the cellular level? Well, remember we had auxin being released from the illuminated side of the plant, and we said that that migrated over to the shade side of the plant. Once we're on the shade side, now we're looking at the cells on the shaded side of the plant, we start our whole new cascade process again. So auxin becomes our ligand. And so when we look at this image here, we have an auxin receptor in our plasma membrane. So here we go, and auxin is going to bind that receptor, and it causes a series of events to occur. One of which is going to be a change in our transcription. So we have growth factors that are going to be produced. Remember, we said that we had cellular processes that were changing as well as genetics. So we have turned on the gene to produce growth factors. We additionally will ask the Golgi apparatus to start making more cell wall material. And the reason for that is we are also going to activate what we call a proton ATPase, or a proton pump. So auxin on this side is going to stimulate these proton pumps to release protons into the extracellular matrix. So for our particular situation here in this cell, our perception is light stimulating the tropin on the illuminated side. Our transduction is that the photoreceptor changes shapes and starts a cascade of events, including the release of auxin. And the response, once this auxin has bound our receptor, the response in these cells is that the cell walls loosen and remodel. And the way that they do this is this release of protons causes these rigid cellulose fibers in the cell wall to break down. And when they do that, in addition to water then entering the cell and causing greater turgor pressure, which then allows those fibers in the cell wall to stretch, and then it's going to be rebuilt, and that is going to cause an overall effect of an elongated portion of the shoot. So here is maybe a more simplified version of that. Here we have our cell beforehand on the left. You can see it's kind of square and squatty. And so our auxin comes in, stimulates the proton pump as the protons are released into the cell wall. Excuse me. It's going to activate actually proteins called expansins that help to loosen that cellulose. And then water is going to flow into the vacuole, and then we're going to start to rebuild that cell wall. And so after that whole process, our cell is much longer. Here's another look at that if you would like to see that in detail. And so here we go. Overall, once we have expanded all these cells on the shaded side of the plant, notice these cells are much longer than these, then our overall response is going to be a differential growth towards the light. So that's how phototropism works. So the second example of cell signaling that I want to talk about involves root gravatropism in plants. Sometimes we call that geotropism. In other words, how does the plant sense which way is down? So the stimulus, or something, a perceived change, for this example is going to be a change in the gravitational direction. So as we see these little plants here, they have been growing straight down, and then we turned them 90 degrees. So now down is in a different direction. So how do the roots sense that in order to have a response so that they can start growing downward in the direction of gravity? Okay, well, our first step, we always said, is perception, or in this case, we also call that gravi-sensing, so sensing gravity, or the perception stage. And so let's just do a quick tour of root tip anatomy. This is just your primary root of a plant and just the very tip of it. And you're not going to need to know all of these details. I'll kind of show you what you do need to know. So there are different zones within the root. We have this elongation zone up here. That's going to come into play later. That's going to be important. Meristematic zone, that just means this is where new cells are being produced. We're not going to involve that right now. But the important cells as far as the perception of gravity is concerned are these kind of orangish rust-colored cells right here at the very tip. And these are what we call the columella cells. They're a part of the root cap. They're called columella cells because they are shaped like columns. And so this image to the right is a light micrograph of these columella cells, and this is in a plant that we call Arabidopsis, and they have 4 different tiers of these columella cells. So you can kind of see right here where those 4 tiers break down. And so for example, in this tier 3, this right here is one columella cell, and here is another one right here. Okay, so actually in Arabidopsis there's a total of 16 columella cells. That's not really that important, I just thought I'd let you know. Okay, so what is important about these columella cells is they contain specific organelles called amyloplasts. And so what that looks like is it's a membrane-bound organelle, and within it are these dense starch granules or starch grains. So you'll see those right here. These are all these amyloplasts filled with starch. And what that means is that they are really heavy. So it makes sense that this is where the root would then be able to perceive gravity. Okay, so here is just sort of a cartoon illustration of that. And so as we started on the left, we have our lovely plant, and it is facing in the down direction. And here are all our amyloplasts. This is just one columella cell, by the way. And so N is our nucleus here, and this vacuole looks pretty small. That vacuole is probably a whole lot bigger, like probably about that big. Okay. In our image here, we have our cell wall, our plasma membrane, and this blue right here actually represents the endoplasmic reticulum. Okay, so pretty quickly, the amyloplasts begin to fall in the direction of gravity as soon as we turn the plant 90 degrees. So even though it says on this image 1 to 2 minutes, keep in mind that those amyloplasts start to fall as soon as that plant hits 90 degrees, and that causes a lot of different things to happen. But this is our perception site. The perception site is the columella cells, and the perception is those amyloplasts falling. So in this particular instance, we don't have a ligand mediated response. This particular perception is a little different. It's a mechanical perception. So that's one of the reasons I wanted to bring this example to you, is just to show you that there are some things that are a little different than the textbook. Okay, so you have to imagine with me, and you may need to tilt your head 90 degrees a little bit. So now we're going to talk about signal transduction. So we have our perception right here. These are our amyloplasts falling, okay, and that starts at time 0. Once they fall, there are all kinds of things that take place. One of the immediate changes is that the pH within the cytosol changes within those columella cells. And I left that kind of vague for a reason, because the columella cells, depending on which tier they are in, that cytosolic pH changes in different directions. So for now, we'll just summarize that as that there are cytosolic pH changes within those columella cells. Okay, and then that also leads to polar localization of what we call pins. Now I've explained that down here. A pin is a transporter within the plasma membrane that allows auxin to go outside of the cell. So what is auxin? Auxin is a plant hormone. So think of that as just another molecule. So you can see this cascading effect. We have amyloplasts falling, changes in cytosolic pH. We have a polar localization of pins. So what that means is that auxins are being taken out of the cell on one side of that cell and not another. Okay, so this is happening over time, and notice our time, we're basically following the dark green in this image. So as we go amyloplast to cytosolic pH to polar localization, we are going further away from this initial stimulus time. Okay, so normally, okay, so that's at the cellular level. So we have effectively had a cascading effect at the cellular level. So inside the cell, we have several cytoplasmic changes that are occurring then. As we said, we had the immediate change in cytosolic pH, and as I mentioned, that differs from one tier of columella cells than the other, and that sets up a gradient of changes in pH across the organ or across the root itself. When those amyloplasts fall, there's pressure on the endoplasmic reticulum, and that actually starts a release of cations. So in this case, it's not a ligand mediated ion channel release. In this case, it is a pressure release on those same ion channels, or maybe not the same ones, other ones. And then if you can imagine, as the weight of these dense starch grains fall, there is a pressure on the cytoskeleton, or even sometimes a breakdown of the cytoskeleton. And keep in mind, those cytoskeletal components are attached to the plasma membrane. They're attached to other components. So when there's pressure on them, then that causes pressure on the plasma membrane as well. So pressure on the plasma membrane, and that can also be related to the next point, which I think is really cool, and sometimes people don't think about this very much. That drop in amyloplasts also causes an electrical potential change across the plasma membrane. So you know how your nerve cells have an electrical gradient, and when you are trying to send information from the brain to your muscles, for instance, there is a change in that potential which causes an electrical current to go across your nerves. Well, this also happens across the plasma membranes of plant cells, and that can travel up the root. So it starts at the cells that perceived gravity, and then it's going to flow up the root, and we're going to talk a little bit about where the growth changes occur. And then because of the pins that we talked about, there is a release of auxin on one side of the cell as opposed to the other. So there's a gradient of auxin being set up as well. So what I wanted to talk about with this is just to kind of show you that it's a little more complicated than just a straight, here's our perception and here's our response all in the same cell. So as we said, the columella cells down here are where change in gravity is perceived. Now, if you've ever seen a root grow and change directions, this is not the area where that growth occurs. It is back here in that elongation zone that I talked about. So this is the area of the root that's actually going to do the changing. So not only do we have a signal transduction response within the cell, now we have to take that signal and bring it up the plant to the area that can actually cause a growth change in order for the root to now grow down. And if you'll recall, we had what we called pins, which allowed for the efflux of auxin from those cells, but in a directional pattern. Okay, so normally as this root tip is growing down, let's say we are going down this way, we have an equal distribution of auxin going outside of these cells because auxin does induce growth. Now, once we have turned our plant on its side, now we have directional efflux of auxin, so there will be more auxin on one side versus another, for instance. So what that means is now we are starting a whole new series of signaling at different cells. So now when we look at this area of growth, these cells in the elongation zone, that plant hormone auxin is going to work as a ligand, and it's going to bind to a receptor in the plasma membrane of the cells within the elongation zone and start a whole new cascade of signal transduction there. So if we look at it like this, we have our columella cells with the pins being activated. So as I said, when our plant is up and down, we have equal distribution of auxin on either side of the plant. Now that we have activated our pins and we have auxin being basically unequally distributed, we have auxin falling towards the side in the same direction as gravity. And unlike our shoots, when we have, what we have in roots is when we have auxin, there's a transient alkalinization of that extracellular matrix, and that reduces elongation. So if we have less elongation on this side and more elongation of cells on this side, then you can see how we would have directional growth in this direction. So in roots, more auxin equals less cellular expansion, and that's how we accomplish our differentiated growth. So we talked a lot about the cytoplasmic responses, and we said that our response side really had cytoplasmic changes. It also has genetic components. So I will not make you memorize these, but I just wanted you to see that within our plant, we do have changes in all kinds of different genes being turned on, turned off, activated, all those kinds of things. So I just wanted to throw that out there as well. So when we put all of that together, our cellular responses work in concert to create an organismal response, so that all these roots can now grow in the direction of gravity so that the plant can be well nourished. So it's really fascinating. Plants don't go anywhere, but they respond to their environment at a cellular level, and then those all come together to create an organismal response. So I'm sure everybody was wondering what happens to plants in space, right? Of course, because that's what everybody thinks of. Actually, I do tend to think of those things. So what does happen to our gravatropic response when we're in in space? Well, our plants do look a little confused. Without any type of gravity, plants do have an internal inherent ability to usually send their roots in the opposite direction of their shoots, so they know that much. They definitely are interested in any kind of stimulus other than gravity. For instance, if you shine a light in one particular direction, the roots will grow in the opposite direction. The roots do tend to seek out where the nutrients are, so where the water is most, they'll grow in that direction. So in the absence of gravity, other factors come into play. So here is some images from space as well. This time it's like growing in every direction because most importantly, the humidity is the same mostly throughout this particular space. So why do we even care what plants do in space? The reason that NASA has done research with that is because if we are going to start looking at sending astronauts, say to Mars, for instance, that's a very long period of time. And so if you're talking about being in space for extended periods of time, where you can't come back to refuel, you're not sending up space shuttles to bring extra materials, then you need to be self-contained within your spacecraft. And so you're going to require food, clean air, some good mental health for our astronauts. And amazingly, plants do provide a lot of that. So we know that plants tend to clean air. They absorb our carbon dioxide that can be toxic to us if it builds up. They do tend to take toxins out of water systems, for instance. And then good mental health. There have actually been lots of studies that show when people are gardening and growing things, that is very good for their mental health. And then, of course, plants are going to provide fresh food, which is much healthier and much tastier than freeze-dried food, for instance. So growing plants in space is going to be critical if we are going to do any long exploratory missions. So now we've gone from cell signaling at this tiny level all the way to traveling in space, and they're directly related. So maybe one of you gets to go traveling to Mars one day. I would love it. Okay, so that is it for today for cell signaling, and I will meet you back here next time.